Thank you and welcome back. Now, as I said, we'll turn to our final session of the day before a brief summary. This is a set of panel presentations and discussions on leveraging mechanisms efforts for M2M2M. Following the talks that we just heard, I think that this will be a really great discussion. I'll now turn it over to Tuuli Lappalainen, core faculty member at the New York Genome Center and associate professor at Columbia, to introduce the panel.
Thanks, Rachel. It's a real pleasure to be able to finish today's fantastic program with a discussion about the middle one of the 3 M's of ICDA, which is mechanisms. And what we will be doing today is that myself, Nicole, and Jay will give a brief introduction into some of the work that we have been doing in the mechanisms working group, some of the vision and the recommendations, and then we will move on to a panel discussion with Jesse, Anna, and Doug, and we will also be taking questions from the audience. But just to get you a little bit oriented into what are we talking about here when we talk about mechanisms in the ICDA context. So this working group is really focused on molecular and cellular mechanisms of disease-associated loci and genetic variants in general. The more sort of physiological end of the mechanisms, that's more in the realm of the medicine working group. And another important emphasis of the mechanisms working group is a focus on generalizable disease agnostic mechanisms. So here we typically would not dig very deep into mechanisms of specific diseases, but rather build generalizable resources to empower this type of work. And this is, of course, an area that has many challenges and a lot of work to do, but also unprecedented opportunity with fantastic technology development, and many of the talks that we have heard today, as well as other work in the community, are fantastic examples of this. And in the mechanisms working group of ICDA, we hope to play a role in accelerating these efforts, and in particular having a role in coordinating efforts and enabling collaborative work. So with that, we'll move on to introducing some of the 3 mechanisms recommendations that we have put forward that are also part of the ICDA white paper. So I'll start first by talking about recommendation number 7 in the white paper, which is human gene regulation map, where the aim is to build a comprehensive map of cis-regulatory elements, of cis-regulatory variant effects, and their target genes. And this is obviously an area that is a major challenge for GWAS interpretation. We all know the challenge of having a non-coding GWAS loci and then having to do a lot of work to be able to just infer even the basics of the potential molecular mechanisms. So here, the vision would be really for people to be very easily able to do easy in silico annotation of high quality assessment of, for example, target genes in cis and enhancer gene targets, etc. Of course, the ultimate goal is to be able to do high quality prediction of the regulatory effects of any variant. It could be a rare variant, any arbitrary variant, which would really kind of, when we get to that level, we would really be able to read the regulatory code of the genome. And then, of course, in addition to the sort of disease-focused analysis, there's also a lot of general biology that we need to understand when it comes to regulatory architecture of the genome, for example, how enhancers work together, how splicing and gene expression contribute to transcriptome variation, etc. These are all areas where there has already been a lot of ongoing and planned efforts that we hope to collaborate with and complement. For projects such as ENCODE, IHEC, Human Cell Atlas, and others have been building a lot of genome annotations of the regulatory elements of the genome. EQTLs and other types of regulatory variants have been mapped and cataloged by projects like GTEx, EBI's EQTL catalog, EQTL Gen, and others. And there has been a lot of very exciting development of experimental assays. Projects like ENCODE have contributed to that. There are upcoming projects from the NIH's IGVF consortium and other work. So there is a lot of things to build on. There are 4 sort of relatively specific areas where we hope that the community would come together to really sort of put an effort on advancing these goals via specific projects or specific sort of coordinated efforts. The first one that we think is a high priority goal is to really take eQTL mapping to cell type level. We've already heard talks about how important this is from many aspects. And we think of this as a single cell GTEx or a population scale human cell atlas. And here also multi-ethnic representation and building those QTL catalogs that extend beyond European ancestry populations is really important. The second aim is kind of building on work that projects like ENCODE and others have been doing, really building, performing epigenomic assays in specific cell types and cell states to annotate these regulatory elements and their target genes, including 3D mapping of enhancer target contacts. The third aim is an experimentally focused effort where we really need to be able to start applying experimental perturbations to measure cis regulatory element and variant function at a very large scale in multiple different cell types and cell states, utilizing technologies such as MPRA, CRISPR, CRISPR-I, CRISPR-A, etc. And then, of course, all of these data needs to come together with computational efforts and with improved computational methods to build better predictions of cis regulatory element and variant effects. And this would ultimately allow extrapolation from the somewhat limited set of cell types that can be analyzed, for example, with in vitro assays, to really kind of build that data into predictions that are applicable ultimately to all cell types. I'll now turn it over to Nicole to talk about recommendation 8.
Thank you, Tuuli. Hi, everybody. So really here, the recommendation 8 is to take in some of the principles of described by Tuuli for 7 and try and advance them to understanding of mechanism of transregulation of gene expression in the human genome. And the stated aim of this recommendation is to build maps of coordinated transactivation in gene expression, particularly when it's driven by transcription factors and other regulators that are turned on and off in different cell states, types, and differentiation pathways. Next slide, please. So the main challenges are summarized here. These are challenges mostly related to understanding how can we reconstruct the cellular networks that underpin a mediated function of disease-associated genes and variants. What are the downstream consequences of perturbing pathways or genes through genetic variation, whether natural or engineered, and indeed what are this type of experiments are really focused on understanding what are the cellular, environmental, and developmental context in which genetic variants exert their effect. So they're really very broad aims, but you know, and potentially also requiring very broad type of experimentation, but will allow us to address the fundamental questions about really not only individual cells, but the cell circuitry. So we identified a number of potential experiments that could fit well under this type of work. We certainly want to focus a lot of attention into the identification and planning of you know, in identifying what are the best reference cellular models that are informative for either key biological processes, physiological processes, or diseases. And indeed, it is unthinkable to do these experiments at the scale of every single cell of the human body, but we really want to identify the best, most informative and viable paradigm that we can use to learn from. And we have a number of experimental assays proposed that include mostly CRISPR screens, but also screening of genetic variation. And so if you could move to the next slide, please. So one of the first thoughts is around how can we exploit genetic variation, different types of genetic variation, what would be most informative. And so we can think of drawing informative subsets of people from the general population, particularly extremes of polygenic risk scores, but also think about individuals that are carriers, with a healthier disease carriers of knockout mutations in genes such as, for instance, transcription factors that can be informative to understand the consequences not only at the cellular level, but indeed at the organismal level. And here again, we want an emphasis on multi-ethnic variation, And we would like to be able to set up experiments where we can sample representative tissues, primary cells, or indeed potentially use organoids or iPSCs derived from these donors to carry out a series of experimentation in vitro, including single cell RNA-seq, ATAC-seq, but also other types of phenotypes that are really informative for understanding regulation in trans, but also cellular phenotypes. The next slide, please. And we can then extend the paradigm that Tuuli has already mentioned before by looking at also the cellular context or the cellular consequences of perturbing genes of variants. And we, again, in vitro systems, we can think of experiments where we perturb the genome. So indeed, we affect the function of the genome through CRISPR-I, CRISPR-E experiments. And this we would want to do in tens of cells exposed to as wide as possible set of conditions. But indeed, also by taking these engineered cells and exposing them to perturbations in vitro, including, for instance, perturbation by either drugs or physiologically relevant stimuli. And really, the goal of all of these experiments is to set up to derive a matrix of, if you want, environment by cellular function, by cell type, that we can use to begin to train our models and to decipher general rules of perturbation in key tissues and systems. And with this, I'd like to pass on to Jay for the third recommendation.
Yeah, so hi everyone, and great to be here and to be a part of this. So recommendation 9, I guess the third of this trio of mechanism-related working group of mechanism-related recommendations is focused on protein coding variants. So next slide. So I guess kind of the premise here is that we're separating coding variants out because they are different, fundamentally than regulatory variants in both challenging and potentially useful ways. So one is just, and this will come up in a few minutes here, I think, is most of the reason for focusing on them separately, just relates from a functional perspective of thinking about variant effects and what the underlying code might be. There's the risk of it being quite different for every protein, as opposed to regulatory variants where you can imagine some shared grammar, so to speak, right? On the other hand, they're more likely to be causal and have large effects than your run-of-the-mill regulatory variant. And then another point worth noting here is that they are finite in number. So if we're primarily interested in common coding variants, just as an example that's come up a number of times in our discussions, if we only look at European populations as an example, there's only about 75,000 of these that are over allele frequency of, say, 0.1%. So the kind of set that you can imagine studying to comprehensiveness or completion. Okay, so the challenges here, I guess there's four kind of the big challenges in this general area. So the first is overlaps with other goals, which is just how do you introduce large numbers of variants of interest at scale into an in vitro model in a way that you can then track in a multiplex assay, right? So that's that kind of there's a broad set of technical challenges around how to introduce variants, but that this is kind of how we'd want to think about doing it in an ideal case. The second and third ones listed here relate to assays, right? So again, related to the challenges that are specific to proteins. We don't even know really the extent to which, quote, generic assays can or can't capture diverse aspects of protein function versus the other extreme, which is where we need to have a boutique assay for every, each of the 20,000 proteins that there are. And then on top of that, there's of course the question of the assay itself, or the mechanics of it. There's the cell line model that you're in, or other in vitro model, and whether or not any given model is actually relevant to the organismal phenotypic consequences of coding variants in said gene. And one is, again, is like, what's the reality? Can we actually get to that point? And then two, what are the practical challenges to actually demonstrating such validity, and how good does it need to be? And then last listed here, I think, at some level that you could argue that the field is, we've squeezed a lot of juice out of the lemon, so to speak, in terms of computational variant effect prediction for protein coding variants. But, we shouldn't give up on further improvements, and, just like protein folding, we shouldn't assume that we've tapped it out completely. And so just can we achieve kind of similar kinds of breakthroughs that take us a major step forward relative to where we are now in terms of just predicting these ab initio. Okay, next slide. So, on the left here is kind of a broad brainstorm of the kinds of projects that might fit under this recommendation, and then I'll talk about a specific pilot, potential pilot that might be a little more focused. So, broad brainstorm, right? So, common complete FIWAS of common coding variants across all available biobanks. So really trying to integrate across everything that's available in more of an association type framework. Second listed here, so isogenic editing at scale, followed of selected variants of interest or large numbers of distributed variants of interest, followed by subjecting these lines to extensive molecular and cellular phenotyping. Saturation mutagenesis, so similar to what our group did on BRCA1 and other groups have done on other proteins, coupled to gene-specific assays, again, getting a little bit away from common variants, but by doing large numbers of variants in individual proteins, even if not all of them are common, most of them aren't common, one still gets at a kind of a distribution of effect size in which to contextualize the effects of individual common variants. And then finally, kind of overlapping with something I talked about on the last slide, just improving computational methods. Okay, so that's kind of the broad brainstorm. And then if we go to the next slide, focusing on a proposed pilot. So just to arc back to something I said earlier, which is just that I still feel like the field does not have a strong handle on how hard this problem is. And so one approach to this would be to pick, on the order of 200 genes that have, that are of strong interest to this community because of the implication of coding variants or particular genes that harbor coding variants that could be causal in common disease, right? But not picking those based on the assayability, which is often what we tend to do when we're looking through our list of candidates for which ones to actually validate. Rather, just focusing on what's of interest. And then really making a hard effort to try to develop a battery of assays that can robustly separate wild type versus loss of function aspects of molecular or cellular function in as many of those 200 genes as possible. And ideally, we would have a limited number of assays that worked for many of those genes, right, that were quote generic or as generic as possible. And then third here, really building out the toolkit. And I think this is where the field feels definitely aligned to make this happen in the next year or two. The molecular toolkit for introducing arbitrary variants of interest into arbitrary models in multiplexable ways. And then finally connecting the dots here and really trying to study larger numbers of variants in these 200 genes across these batteries of assays to really build out a database of effects, but also know-how and just grounding in terms of really how hard this problem is going to be to scale from 200 to 20,000 genes. Right? Okay, I will stop there. And do we want to, Tuuli, you want to take it from here to lead up the panel discussion?
Yeah, I guess I can just I can just briefly introduce our panelists. So we have Gloyn from Stanford, who already gave a talk today about some of the very interesting work, probably most closely related to the cellular mechanisms work that Nicole outlined. Jesse Engreitz from Stanford works on enhancer biology and cis gene regulation. And then we have Doug Fowler from University of Washington, whose work focuses on protein coding variation. Nicole, I don't know if you want to also discuss some of the sort of general questions or the kind of perspectives that we ask ourselves all the time regarding some of the generalizable challenges in the field.
Yeah, so we really would like to have the rest of the discussion to be an interactive one. So we'll be reading your question both on the Slack and also on the website. And we have a set of questions that we would like to discuss, but also the broad topic is really about hearing from you what you feel are the key scientific questions for each recommendation that we might want to address in perhaps more specific ways. Whether there is already related work that is already ongoing that you want to flag to our attention, whether here or later on. And we're really interested in hearing from the community what's already going on, as well as what people want to do. And obviously this goes from a smaller scale effort all the way to larger initiatives. Really, part of this discussion, this is the beginning of the discussion, to really identify what are the gaps and also perhaps the barriers to achieving some of these goals, and the kind of things that really require focused effort by the community, and perhaps to guide, as we mentioned, some of the initial relevant pilot projects we might want to put forward, similarly to what Jay has mentioned for the last recommendation from the community. So perhaps we could, I've seen there's a couple of questions already, so maybe you want to, there were a couple of questions for Tuuli that we can maybe take forward and bounce to our panelists. So yes. The first, there were a couple for Tuuli, but I guess in general also for recommendation 7, and I would like to also call Jesse to respond to this. But so are there plans to include the dimension of development and aging in generational gene regulation maps? Because patterns might differ within a cell type across development. And the other one is, again, regarding the regulatory map, would you consider including chemical perturbations that are relevant to various cell types, such as chemical mediators of inflammation, sex hormones, FDA, small molecules, etc., and not just genetic perturbations? So I don't know if Jesse, you want to take this question, or Tuuli, want to go first?
Sure, I can take it. I think this is exactly one of the major challenges of building a human gene regulation map, which is that the regulatory code is different in every cell type in the body and at different developmental time points or in response to different stimuli. It's really quite incredible to think about a 200 base pair enhancer sequence could have trillions of possible sequences that has a different function in every one of the thousands of cell types or states in the body. So how do we get a handle on that vast complexity? I think that we heard today about some really exciting opportunities for tackling that problem that might actually be able to address these, temporal or chemical perturbation specific effects. What we really want is some sort of computational model or lookup table where we could plug in a variant given some particular cell type and state and predict the effect on gene expression. And so how do we get to that point? I think, as Tuuli mentioned in the recommendation here, it's, I think we can start to think about, well, can we integrate CRISPR perturbations in a few cell types and deep single cell eQTL maps in perhaps the same cell types, and figure out the rules of gene regulation, that we could apply to single cell maps. So perhaps someday in the future, we'd be able to do single cell ATAC-seq in every one of these developmental time points or in cell types exposed to many different perturbations. And from that data, apply all of this knowledge from a few cell types that we learned with perturbations and actually create the full map in any cell type that we want to. So I think it's really exciting for us to think about, what are the initial pilot projects and next steps we need to do to get to that point.
I'll maybe add one thing, that when it comes to environmental interactions, like if you think about the genome that is sitting in the nucleus and trying to sort of like decide whether to bind some transcription factor, whether to activate gene expression or something, the immediate environment for that piece of DNA there is the nucleus and the cell. So I think that when we think about how, for example, age or sex or drugs or whatever affect gene environment interactions, we need to think about how do they affect cellular state. And that's kind of like, at least in my thinking, the key to understand how these things work.
And perhaps if I could also extend this question to elaborate a little bit more. It seems like obviously the availability of informative training models is really important. And so the definition of what is training models will be really critical. And so I don't know whether, Jesse, you want to maybe, elaborate a little bit more in terms of, how do we approach breadth, for instance, versus depth? Where should we start? What is, assuming we can do everything immediately.
Right. Well, I mean, I'll just say something specific just to get us started. I think one of the key things as a community we need to do is define some of the prediction goals. Like you said, what is the prediction problem? And maybe there are a few that are relevant. Here are two. One would be, given any non-coding variant in a given cell type, can you predict the quantitative effect on the expression of every nearby gene in cis? And maybe a second, slightly different problem would be, given a non-coding GWAS variant that you know is causal, can you tell me the cell type and the gene that's relevant? So I think we could start to define those problems in a relatively specific way. And then we have to think about what are all of the variables and parameters and measurements that we want to collect to inform those problems. Could we choose 10 cell types that we stimulate in various ways and go really deep and find the top 50,000 cis-UTL signals and perturb every one of the 20,000 or so distal regulatory elements in that cell type, and use that as training data to answer these questions? I think we could start to make and state goals of that type, and building on that, then say, well, okay, are those predictions that we can create, do those hold up in cells when we look across individuals of different sex, or if we perturb the cells in different ways, or if we look in cells from different ancestry groups? And I think, and so I think that I think those types of experiments are now possible. We need to push on the technologies to make it, but also at the same time we need to help to build this community. Can we generate all of these data sets and put them together in the same place, in the same annotation framework, in the same computational spot, so that we can assign to our computational undergraduates as a problem to solve gene regulation using this data set. I think there are both technology and community problems that we can start to think about and solve.
So maybe you can skip to recommendation 9. There was a question by Jeremy about how much would a perfect prediction of protein folding improve our ability to predict coding variant effects on pathogenicity, phenotypic effects. So maybe Doug, you could take this, and Jay can elaborate further.
Sure. I guess the question is just really equivalent to, like, if we had a structure for every protein, how much would that help? The answer is, I think, a lot. If you look at even existing protein variant effect predictor performance, first of all, more or less, or a lot of them anyway, use the protein structure and things learned from the protein structure as features to make the predictions, and those predictors tend to perform, when they have those features, much better. So I think I think it would help a lot if we had structures. With that said, if the goal is to get to clinically useful inferences of variant function, then again, if you look at the sort of current generation of variant effect predictors, even in the presence of structures, I would argue that in general the accuracy is not high enough that you could convince a clinician to use that data as strong evidence, those predictions as strong evidence to like interpret the variant as pathogenic or
But
benign. But for things like prioritizing, variants to test further, having protein structures would be really stellar. That I think the DeepMind paper has stimulated a lot of thoughts in this space. Of course, it performs. It also performs much better when it has a homology model to work from, and without a homology model, though it does much better than previous predictors, it's still not perfect. So
I mean, Doug, would you agree? It's arguably the case that the bar is lower here, right? For relative to the U.S., where you're going to be using it clinically and you need to be right here. You're just, if you can predict it 90% of the time, right, correctly, that's pretty good, right?
Yep. Well, I mean, every iota by which you improve your prediction efficacy, right, reduces the amount of additional experimental work you'll need to do, because your prioritized list will get that much better, right? So I agree with you.
Maybe just ask you, I don't think this is on the audience question list, but kind of a question I'd love to hear your thoughts on. So, I mean, how hard do you think this assay problem is going to be, right? What are the major challenges? And then if you had to guess, right, these 200 not arbitrarily selected genes, but genes selected for reasons other than anything having to do with their perceived assayability, what proportion will require boutique assays versus being amenable to these more generic assays, particularly, again, if the goal is not 99.95% clinical accuracy, but rather the goal of prioritizing functional variants for fine mapping and for understanding, GWAS hits.
Mm-hmm. Yeah, those are both great questions. I think I guess to start with the second one, I think if we just want an assay, I would venture to say that the vast majority of those 200 will, at least in principle, have either a protein binding, like 2 hybrid type assay, or a protein stability type assay that would work, right? And then I think the relevant question becomes, is that data actually going to be useful, right? Which I think for some that is, will data from those sort of generic assays that we have in hand today be useful? And I think for many of the 200, the answer will be yes, right? And we can prove that by comparing especially known clinical variants, to see how they perform. But I think in a reasonably large number of cases, probably many but not most, we'll run into the same problem that was discussed relative to recommendation 7, which is that sure, a protein stability assay for, say, a sarcomere component may be highly relevant in the right cell type, but in the absence of the right cell type, that assay might not really work, right? So we might have an assay for a lot of those genes, but it might still take a lot of work to get that assay into the right cell context to make it informative in the way that we would like it to be.
Yeah, let me continue on that note and pass it on to Anna. So your talk and the work that you've been doing is a beautiful example of how you kind of move from those molecular mechanisms to sort of cellular, cell type specific mechanisms, and also get physiological insights into a specific disease out of that. But like, given how much work it took for type 2 diabetes, what are the kind of some of the lessons that you have learned that might help us to accelerate building that kind of a machine of discovery for any disease or any cell type or let's say tissue or organ system?
Yeah, no, great question. You're absolutely right. If there's anything I've learned over the last 5 years is just how much effort goes into, first of all, being confident in the cellular model that you're going to use for these studies, and then the enormous amount of effort that goes into that cellular assay, even before you start trying to work at scale. I think one of the things that really excites me about ICDA and the opportunity we have as a community is the fact that we can share some of this burden in a way that we bring together the tools that are required to take these assays that take a long time to establish, taking the toolkits that are required, and when something works, being able to work with another group that has expertise in a different cell line where appropriate, and being able to sort of extend your reach in an efficient manner. You saw, those of you who listened to my talk will have seen at the end some of the efforts that we're going to try and link up with others who have interests within diabetes so that we can extend our reach. So things that we're simply doing within the diabetes space is trying to be smart about the libraries that we put together for the targeted assays at least, so that we can, whatever we've assembled for one cell type, we can roll out into another cell type and be efficient in that manner. I'd like to just sort of also link back to what Jessie was saying about opportunities here as a community with the different recommendations we've got here and the different types of assays. I also see opportunities for us, if we're smart at this point with the types of cell types that are selected, the assays that are chosen, that we could down the line bring these data sets together in a way that we could only at the moment dream of doing, so that we can understand things from the SNP to the regulatory effect to the cellular phenotype. Because ultimately, even if we have understood the effect of a SNP on gene expression, what we really want to know as well is whether those are disease-relevant phenotypes at the end of it that come out of it and are related to the biological process, if we're going to move it forward for a medicine. And I think putting a framework together where we're working on a similar scaffold will make this so much more achievable.
Very good. So I'd like to take a question from the Slido. This is from Abhi Ratnakumar. So he's asking, is single-cell ATAC-seq the gold standard for building these high-quality regulatory and answer maps? What is the best technology for building these maps? So perhaps Jessie would like to maybe elaborate a little bit on this.
Sure. Was the question about single-cell RNA-seq combined with CRISPR perturbations? Is that the question?
ATAC-seq. So he's asking whether ATAC-seq is the gold standard for building these high-quality regulatory and answer maps. Or...
Yes, right. Well, I think that's a question that we need to answer as a community. The question, I guess, is what is the minimal amount of data that we need to collect in a given cell type and state in order to predict everything else? And at the moment, we have some interesting models that are starting to show a lot of promise. And David Kelly told us about some earlier today that involve collecting ChIP-seq and DNA-seq and ATAC-seq data. And so I think it's a really important computational challenge for us. Can we make the same types of inferences from only single-cell ATAC-seq? Do we also need 5' RNA single-cell RNA-seq so we can find the exact transcript start sites, promoters? Do we need some sort of single-cell ChIP-seq data? I think as we begin to define these specific computational challenges and set up positive control data sets from CRISPR and from ETLs, we can start to answer those questions. But I think for now we should certainly encourage folks to generate these sorts of data sets in their favorite cell types, because they are even now very useful.
Okay.
Yeah, I could ask a slightly sort of a practically oriented question from all of you. So when it comes to, I think one of the bigger challenges that we have in the sort of functional genomic space is bringing these data together and actually organizing it smartly, because this is sort of, I always say that genetics is kind of easy. You either have a variant or not, but we all know that anything that we measure, it just doesn't work like that. So what do you see as some of the major needs and challenges regarding data integration and dissemination to the community? I don't know who wants to start. Maybe Doug.
Sure. I think that question has maybe two layers of answers. One is that, for each type of assay as it emerges, or each of these major groups of assays, we really need to do a good job of defining what the reporting standards are for the assay, and then enforcing those reporting standards. And in my sort of section of this field where we're doing saturation mutagenesis of proteins, that's a mess. We've only just started that, and it makes comparing data sets, even of ostensibly the same type, very hard. Others of you are in much better shape. But I think we absolutely, have to not only have those standards articulated, but really enforce them strongly. As a community. And then I think the second thing that we really need to be able to integrate now, more and more disparate data types is a really strong and clear and computable ontology, right? That explains, because I don't think we're going to end up in a situation where we pick 3 cell lines, like K562 and whatever, and then we just do all the assays in the same cell lines, right? Instead, we're going to need a really clear ontology that allows us to sort of fairly compare and integrate even when experiments of very different types have been done, perhaps in different contexts.
Doug, for clarity, you mean an ontology of cell types and ontology of assays? What kind of ontology do you mean?
I think both of those things, right? An ontology of assays, but also, descriptions of context and all that.
Yeah, Anna and Jesse, do you have anything to add to that from your perspective?
I think Doug, hit some of the major challenges there. One that we've been thinking a lot about recently is, related to having access to the original data and pipelines that were used to generate and analyze the data, both for reproducibility, but also because sometimes we want to extract something from the data sets that the original data producers weren't, didn't intend to create. One example is having an assignment of power for every single EQTL association test that is done for every variant in every gene. Right now, we have the set of positive examples where there was a statistically significant genome-wide association between a variant and a gene. But we'd also like to have negative examples where we're fairly confident that this variant doesn't affect gene expression. And so it would be, in order to do that computation is actually quite expensive, it turns out. And we have to think about how do we, pull all of the raw data and pipelines into the same place so that we could extract these, new types of information as we realize that they could be useful.
I would just add, I totally agree that it would be fantastic if the field could find ways of implementing similar guidelines that we can now take a standard for the sharing of genetic GWAS data and sequence data. I'm not sure what those are yet, and I think that's something that the community needs to work on. But it would be a sensible place to find ourselves in, where we could have a summary way of sharing data for those that want to just do lookups, and then a way of accessing the raw data for those who want to go back and do their reanalysis. And I would echo what Doug had said about having a set of guidelines and recommendations on how data is shared and how experiments are put together, because I think that would be enormously helpful for the community, and to do it sooner rather than later, before people invest in these extraordinarily time-consuming and expensive endeavors.
Very good. There's a lot of still questions that we won't be able to address, but I suggest we take them on Slack. But maybe a very, very final one for Anna. So there's a couple of questions on the slide about the fact that we're talking about coding and non-coding variation somewhat separately, but there's obviously a lot of scope for also looking at haplotype effects or interactions. So do you think we should worry about them now, or should we first focus on nailing separately coding and non-coding variation? And from your experience, what is there to be learned from the two?
I think it's a really interesting question. In some ways, the attempts that we're making at the moment with doing CRISPR knockout screens aren't really looking at regulatory effects. They're looking at what happens when you have a protein truncating variant or equivalent within that gene. So I think some of what we're doing at a kind of gene level has got implications for protein coding variants. I think definitely when we start to work with the CRISPR-I and CRISPR-A and doing enhancer screens, those are a separate line of inquiry. I'm really excited myself about some of the opportunities we might have with the type of experiments that Doug and Jay have been describing, where we've got genes that we know that there are common regulatory variants in or common coding alleles in, about doing MAVES on the backbone of those alleles, so we can start to see some of the effects of regulation on cellular phenotypes. But of course, you know, there's always more we can do, and where would you put that in the pecking order in terms of your priority? But perhaps I would argue that once you have an assay that's up and running and you've made the investment in it, if there's good biological reasons to explore those effects, the reward would be pretty good, given that you've put all the hard work in already with the assay.
I could probably very briefly add that I think that we make a mistake as a community if we kind of put coding and regulatory variants and common and rare variants very distinctly in separate bins and just kind of like analyze them in completely separate communities. There is a lot of shared biology that we just need to address.
Totally agree.
Very good. I think it's a great panel discussion. We need to close here and give the word back to Cecilia. Thanks all very much to all the panelists. We will be trying to address the remaining questions on the Slack. So thank you everybody, and Cecilia.